Imagine if your body worked like a Lego set. Break off a piece and you could simply snap in a new one. In reality, nature has already given us something remarkably similar in the form of stem cells. Unlike ordinary cells, they start out blank, with the extraordinary ability to transform into almost anything. Bone, muscle, neurons, even parts of whole organs. They're kind of like the body's master builders. This incredible flexibility makes them one of the most exciting tools in modern medicine. Around the world, scientists are learning how to harness them to repair damaged tissues, treat chronic diseases, and perhaps one day grow entire replacement organs on demand. In this presentation, I'll walk you through three areas: the origins of stem cells and how the field has progressed, the challenges, costs, and ethical questions they raise, and the possibilities for the future of cell and tissue engineering. Stem cells first burst into the public spotlight in the 1990s, and almost immediately they became a scientific marvel wrapped in controversy. Researchers isolated human embryonic stem cells, cells taken from very early embryos that could develop into any cell type in the body. For scientists, it was like having a blueprint for building life itself, but for society, it raised ethical questions, since creating these cells involved destroying embryos. In response, researchers turned to what nature had already hidden inside of us: adult stem cells. Your bone marrow, for example, contains stem cells that constantly regenerate new blood cells. This was far less controversial. As a result, bone marrow transplants have been saving lives for decades. But there was a catch. These cells were limited. Adult stem cells could only form certain cell types. They couldn't match the flexibility of embryonic stem cells. Then, in 2006, came a discovery that completely rewrote the field. Japanese scientist Shinya Yamanaka showed that you could take an ordinary adult skin cell and rewind it back into a stem cell state. These induced pluripotent stem cells, or iPSCs, required no embryos at all. It was like discovering biology's reset button, and it won him a Nobel Prize. Overnight, the field exploded with new possibilities. Science fiction often imagines futuristic medicine, growing new skin, repairing broken bones, even building organs in a lab. But today, stem cells are turning that fiction into reality. Take burn victims, for example. Traditional skin grafts rely on taking healthy skin from another part of the patient's body, which is painful and limited. However, today scientists can grow lab-made skin grafts using the patient's own cells. They start by cultivating keratinocytes, the cells that form the outer layer of the skin, into a thin sheet. This sheet is then combined with a dermis-like scaffold, a supportive layer that mimics the natural structure of the skin. Finally, the graft is placed onto the wound, resulting in a safe, natural, and personalized skin replacement that integrates seamlessly with the patient's body. Beyond the skin, stem cells are also being used to repair bone and bone marrow. Scientists place skeletal stem cells onto a three-dimensional scaffold, which provides structure and encourages the growth of blood vessels. This environment helps the stem cells develop into different types of cells that are needed to form a functional bone marrow organ. This approach could one day help treat fractures, bone loss, and blood-related conditions, offering new possibilities for patients who currently have limited options. And in the research lab, stem cells are giving rise to organoids, which are tiny simplified versions of organs. Miniature hearts that beat in a dish or clusters of brain-like tissue, used to model neurological diseases like Alzheimer's, can now allow scientists to study complex human biology without experimenting directly on patients. Despite their promises, however, stem cells don't always behave the way we want them to. If they are grown too quickly, they can form cancerous tumors. Transplant them and the immune system might reject them. And even if they do work, scaling up is incredibly difficult. What's easy in a small petri dish doesn't always translate into making a full-sized functional organ. It's like trying to take your grandmother's cookie recipe and mass produce it in a factory. The ingredients may be the same, but the process rarely scales perfectly. Then there's the cost. Current stem cell treatments, when they are available, can run into the tens of thousands of dollars per patient. The challenge is figuring out how to make these therapies safe, reliable, and affordable on a global scale. From the very beginning, stem cell research has sparked ethical debate. Early work with embryonic stem cells raised moral questions about the destruction of embryos. Even today, with iPSCs avoiding that issue, new dilemmas have emerged. One of the biggest is equity. Who actually gets access? If lab-grown organs or stem cell therapies remain so expensive that only billionaires can afford them, are we widening the healthcare gap instead of closing it?